Hello, everyone. I am Dr. Alper Tunga Özdemir. First of all, I would like to thank the scientific committee for giving me this opportunity. Today, I will present you our study on the detection of B-cell producing IgE type antibodies against the house dust mite allergen DERP1. House dust allergy is an allergic reaction that develops in response to allergens such as DERP1, which is primarily found in dust mites inhabiting indoor environments. DERP1, a major allergen produced by dust mites, triggers immune response in allergic individuals. Exposure to DERP1 leads to symptoms such as nasal congestion, sneezing, coughing, itching, and watery eyes. The basic mechanism in the emergence of symptoms in allergic disease can be summarized as the binding of allergen-specific IgE antibodies to mast and basophil cells, and the sudden release of molecules with high biological activity such as leukotrienes, histamine, and bradykinin. The primary approach to preventing allergic reaction is avoid the exposure to allergens, although this might not always be feasible. In situations where allergen exposure cannot be avoided, symptoms can be managed using antihistamines, steroids, and biological agents. Each treatment method has its own advantages and disadvantages, and sometimes treatment may be ineffective or results in side effects may impact the patient's quality of life. There is a need to develop targeted approach to prevent these unwanted situations. Currently, there are antibody-based treatment approaches that block the interaction of the IgE molecule with its receptor Fc epsilon R1. Unfortunately, this approach not only blocks allergen-specific IgE molecules, but also has off-target effects affecting all existing IgE molecules. Additionally, it does not affect the B cells, which are the source of the problem. Therefore, when treatment is discontinued, IgE levels rise again and symptoms come back. B cell receptors, or BCR, consists of a complex of antibodies uniquely expressed by B cells and CD79A and CD79B molecules responsible for the signaling transmission. The part that binds the antigen is defined as variable region, while the part embedded in the cell membrane is defined as the constant region. Each B cell can be specific for a different epitope of the target antigen. This means that multiple clones of B cell binding to the same antigen are formed. Polyclonal B cell response is an effective solution for eliminating antigenic structure, but it complicates the identification of target B cell in allergic response formation. Identifying allergen-specific B cell and designing the treatment approach targeting them will contribute to a better understanding of the disease and the development of effective targeted therapies. Therefore, we aim to develop an approach that could detect B cell producing IgE antibodies against DERP1. For this purpose, peripheral blood mononuclear cells, or PBMC isolation, was performed by taking blood samples from 7 female and 3 male patients diagnosed for the first time with DERP1 positive by skin prick test. B cells were then isolated using Rosette-Sepp human B cell enrichment cocktail. For activation, anti-human CD40 monoclonal antibody, interleukin-2, interleukin-10, and interleukin-4 cytokines were added to medium according to B cell activation protocols. To detect DERP1 specific B cell, we used mimotope peptide antigens previously described in the literature and prepared streptavidin PE labeled tetramers. Then flow-cytometry analysis were performed. To evaluate B cells, CD19 antibody labeled with FITC and PE labeled MT1, MT2, and MT3 tetramers were added respectively. HER-CP 5.5 labeled anti-IgE antibody was added to the same two tubes to identify IgE producing B cells. Flow-cytometry analysis was performed with the following gating strategy. First, singlets were selected via FSCA, FSCH, Lymphocytes were then selected via SSCA, FSCA, and FSCA CD19 FITC was used to select B lymphocytes. Among CD19 positive cells, the frequencies of IgE, MT1, MT2, and MT3 positive cells were determined respectively. In our statistical analysis, there was no significant difference between B lymphocyte frequencies were compared, but when CD19 and IgE positive cell frequencies were compared, we found that IgE positive B cell levels were significantly higher in the patient group. For DRP1 specific pneumothorax desmosomes, we found that MT1, MT2, and MT3 positive B cell frequencies were significantly higher in the patient group compared to the control group. As a conclusion, we were able to detect DRP1 specific B cells by Flow-Cytometry method. DRP1 nematodes were able to bind the allergic B cells, and this binding was significantly higher compared to the healthy individuals. This approach seems to be a promising approach for the diagnosis and the treatment monitoring of DRP1-related allergic diseases. Our promising study data need to be validated with the larger patient groups. This is my final slide of my presentation. I would like to thank you for your patience and goodbye.